Dear students, today we will learn about extracellular structures. Let us begin with the introduction. Although the plasma membranes serve as the main boundaries of cells, many types of cells are surrounded by an external layer of secreted macromolecules which may exist in the form of cell walls or extracellular matrix. A rigid cell wall commonly surrounds the cells of bacteria, fungi, algae and higher plants. Cell wall is made up of cellulose in plants and some fungi, silica, calcium carbonate, agar and kerosene in case of protease and algae, or chitin in case of fungi. While in animal cells, a cell wall is usually absent, but the cells in tissues have an extracellular matrix composed of proteins and polysaccharides. In addition to providing structural support to cells and tissues, these extracellular structures also play important roles in multicellular organisms. Let us now see the extracellular structures one by one. Let us start with the prokaryotic extracellular structures. First, let us take up the cell wall in prokaryotes. Prokaryotic cell walls are composed of mainly peptidoglycan in bacteria. Depending upon their ability to take up gram-stain, bacteria are grouped into two types, gram-positive and gram-negative. Cell wall in gram-positive bacteria is thick and made up of many layers of peptidoglycan and teichoic acids. While in case of gram-negative bacteria, the cell wall is thin and consists of a few layers of peptidoglycan surrounded by an outer lipid membrane containing lipopolysaccharides and lipoproteins. On the other hand, the cell wall of archaebacteria lack peptidoglycan, and four types of cell wall are commonly found in archaebacteria. The first one is, in some methanogens such as Methanobacterium and Methanothermus, cell wall is composed of pseudopeptidoglycan, also called pseudomurin, which bears some resemblance to peptidoglycan. The second type of cell wall is composed entirely of a thick layer of polysaccharides found in Methanosarcina and Helococcus. The polysaccharides in such type of cell wall may also be sulfated, as in the case of Helococcus. The third type of cell wall in archaebacteria consists of walls made up of glycoproteins. For example, in Halobacterium, which is a hyperthermophile, and some methanogens. The cell wall proteins in Halobacterium have a high content of acidic amino acids, which gives negative charge to the wall and is stabilized by positive sodium ions present in the surroundings under natural conditions. This explains why Halobacterium thrives only under high salinity conditions. In the fourth type, the cell wall in other archaebacteria such as Methanomicrobium and Desulfurococcus are composed of surface layer proteins known as S layer. The next structure is the glycocalyx. The glycocalyx, also called capsule or slime layer or extracellular polysaccharide, is a gelatinous polysaccharide and or polypeptide covering lying externally to the cell wall. The exact chemical composition varies depending on the species. Capsules may protect pathogens from phagocytosis and enable adherence to surfaces, prevent desiccation, and may provide nutrients. In addition, many prokaryotic microorganisms may form biofilms complex grouping of microorganisms held together by a sticky protective coating secreted by the cells. The next structure is the flagella. Flagella are long filamentous appendages consisting of a filament, hook and vessel body and help to push the cell by rotating. The filament is composed of the globular protein flagellin, which is arranged in several intertwined chains that forms a thick helix around a hollow core. Motile bacteria move by taxis in two ways: positive taxis or movement towards an attractant and negative taxis or movement away from a repellent. The next are the axial filaments. Axial filaments are similar to flagella except that they wrap around the cell and spiral cells that move by means of an axial filament or endoflagellum and are called spirochetes. Next are the fimbriae and pili. Fimbriae are short, thin appendages. Fimbriae are usually many and help the cells adhere to surfaces, while pili are only 1 or 2 per cells and help join cells during conjugation for the transfer of DNA as sex pili, or are used for special types of movement like twitching in Pseudomonas aeruginosa, Neisseria gonorrhea and some strains of E. coli, or the gliding movement of myxobacteria. The next structures are the endospores. Endospores are tough spores formed by some prokaryotes under unfavorable conditions. Endospores are resistant to ultraviolet and gamma radiation, drying, high temperature, starvation and chemical disinfectants, and they ensure the survival of the DNA under stressful conditions. Next, let us take up the second type of extracellular structures, that is the plant cell walls. The cell walls of most algae and higher plant cells are 0.1 to several micrometer in thickness and composed principally of cellulose, which is a linear polymer containing more than 10,000 glucose residues. The glucose residues in cellulose are joined by beta-1,4 linkages, resulting in the formation of long straight chains of polysaccharides. Many such chains of polysaccharides associate with one another in a parallel manner to form cellulose microfibrils. Plant cell walls are generally made up of 3 strata or layers. The first one is the primary cell wall. It is a thin, flexible and extensible layer formed when the cell is still growing, and this layer is generally made up of mostly carbohydrates such as cellulose, hemicellulose and pectin. The cellulose microfibrils are linked by hemicellulosic linkers, and these cellulose hemicellulose networks are embedded in the pectin matrix. The main hemicellulose in the primary cell wall of plant cells is xyloglucan, except in grass cell walls where it is replaced by glucurona arabinoxylon. In most plants, the outer part of the primary cell wall is impregnated with cutin and wax to form the cuticle. The second layer is the secondary cell wall. In some cells, such as the conducting cells in xylem, a thick secondary layer is formed inside the primary cell wall after the cell is fully grown. Secondary walls gives mechanical strength and modify permeability of plant cell walls. Wood is made up of mostly secondary cell walls and is commonly composed of cellulose, xylem, which is a hemicellulose, and lignin, which is a complex phenolic polymer. Lignin is present in the spaces between cellulose, hemicellulose and pectin in the cell wall and strengthens the cell wall as well as waterproofs it. In addition, most plant cell walls also contain glycosylated structural proteins which forms 1 to 5%, such as hydroxyproline-rich glycoproteins, arabinogalactan proteins, glycine-rich proteins and proline-rich proteins. The secondary cell walls of the epidermis and the Kasparin strip in the endodermis of roots and cork cells contain cutin and suberin respectively, which are polyesters that forms a water impermeable barrier in the cells, while cell walls of grasses contain microscopic silica crystals which protects from herbivore grazing. Additionally, plant secondary walls may also contain numerous enzymes such as hydrolases, esterases, peroxidases and transglycosylases that function to cut, trim or cross-link the polymers in the wall or stored carbohydrates, for example, nasturtium in endosperm cell walls in the cereal seeds and glucans and other polysaccharides of other seeds which are digested to provide nourishment to the growing embryo during seed germination. The next layer is the middle lamella. It is the outermost layer rich in magnesium and calcium salts of pactic acid. This forms the interface between adjacent plant cell walls, while cell-to-cell communication occurs through plasmodesmata, which interconnects the cytoplasm of adjacent protoplasts through the secondary cell walls. Next are the fungal cell walls. The cell wall forms an outer layer surrounding the plasma membrane in fungi. Unlike plant cell walls, the cell walls of most true fungi lack cellulose. The cell walls of true fungi have three main components. The first one is chitin. Chitin is a polymer consisting of unbranched chains of beta-1,4 link and acetyl glucosamine in Ascomycota and Basidiomycota, or poly beta-1,4 link and acetyl glucosamine or chitosan in Zygomycota. The next are the glucans. These are glucose polymers that cross-link chitin or chitosan polymers and consist of beta-glucans, while glucose molecules link via beta-1,3 or beta-1,6 bonds that provide rigidity to the cell wall, and alpha-glucans with glucose molecules link via alpha-1,3 and or alpha-1,4 bonds that forms part of the matrix. The third components are the proteins. Proteins present in fungal cell walls include enzymes required for cell wall synthesis and lysis, as well as structural proteins which are mostly glycosylated and contains mannose called mannoproteins or mannans. Next, let us take up other eukaryotic cell walls. The cells of many algae have a covering of exopolysaccharides, while the diatoms have a surrounding layer of silica called frustula. Other organisms like the foraminiferans, tested amoebae and silicoflagellates also have an external covering made up of minerals around their cells. On the other hand, the cells of many green algae, some red algae also secretes a skeleton of calcium carbonate around their cells, while some golden algae, ciliates and choanoflagellates produce protective cell-like outer covering called lorica. Similarly, some dinoflagellates have a covering of cellulose plates called theca, and coccolithophorids have coccoliths. In Oomycetes, also known as water moles, the cell walls are made up of cellulose and glucans, except in some genera such as Achlea and Saprolegnia. Therefore, although they are saprotrophic plant pathogens like fungi, they have been reclassified as heterocones along with related autotrophic brown algae and diatoms due to the absence of chitin in their cell walls. Next, let us see the extracellular matrix. Animals and protozoan cells lack cell walls and instead secrete a mass of carbohydrates and proteins in the intercellular spaces between adjacent cells called extracellular matrix, which helps in maintaining the shape of cell and also provide structural stability to the cell. The extracellular matrix is composed mainly of long fibers of the protein collagen modified with carbohydrates called collagen fibrils. Collagens are a family of about 19 different proteins and they form characteristic triple helices with 3 polypeptide chains wound around one another in a rope-like structure. The sequence of amino acids in the triple helix domain of collagen consists of gly, X and Y repeats in which gly is glycine, X is frequently proline and Y is usually hydroxyproline. The collagen fibers are interwoven with carbohydrate-bearing proteoglycans and both collagen and proteoglycans are attached to proteins in the extracellular matrix such as fibronectin proteins. The fibronectin proteins in turn interact with integrin proteins which are transmembrane proteins present in the plasma membranes of eukaryotic cells that lack cell walls. On the cytosolic side, the integrin proteins are linked to the cytoskeleton and thus they anchor the cells to the extracellular matrix and also helps in triggering signaling pathways in response to both chemical and mechanical signals from the extracellular matrix. The basal lamina or basement membrane are a type of extracellular matrix and is composed of a thin sheet-like layer on which the epithelial cells rest. Basal lamina also surrounds muscle cells, adipose cells and peripheral nerves. In addition, extracellular matrix is also found in connective tissues, example in bones where they are hardened by deposition of calcium phosphate crystals, tendons containing high proportion of fibrous proteins and cartilage containing high concentration of polysaccharides and the loose connective tissues beneath epithelial cell layers in which fibroblasts are distributed. The extracellular matrix helps in protecting the cells within tissues from external stresses and also helps in transmitting signals from the outside of the cell to the inside of the cell. In addition, the extracellular matrix may also serve as the site of attachment for microbial pathogens during an infection. For example, the bacterium Streptococcus pyogenes causing strep throat and other infection binds to fibronectin in the extracellular matrix of the cells lining the oropharynx or the upper region of the throat. Let us now see the conclusion. Cells of many organisms contain different extracellular structures, including cell walls and extracellular matrix outside the plasma membrane. These extracellular structures have different structures and varied compositions in different cells, tissues, or organisms. The cell walls and extracellular matrix serve many important functions in addition to providing protection and mechanical support to the cells. Thank you.